The following is a conversation with Professor Karen Scott, who holds a professorship at the Rowett Institute at the University of Aberdeen, one of the world's leading research centres for gut health, gut microbiome, and all things microbial. What you can expect to learn on today's podcast is: what are probiotics? Does Professor Scott take probiotics? What are some of the limitations of current microbiome research? What is a healthy microbiome? And how can you make yours healthier through diet, nutrition, and potentially supplements? This was a really fun conversation. I was interviewed a couple of times by Professor Scott, which was a nice change. And we also had a fun debate at the end, which was totally impromptu. And we'll release that as a separate clip. And yeah, I had a lot of fun recording this one. This is at a good level for all of the listeners. You could listen to this as a professor or someone very, very deep into the subject matter and still learn things. Likewise, this could be your first ever Inside Matters podcast and you'll learn a heck of a lot. As always, if you're enjoying the podcast, please hover your finger over to that like and subscribe button and kindly consider giving us a 5-star review. It is absolutely the best way to support the podcast. We are gaining some pretty decent traction, which I'm really excited about. That means that lots of people are enjoying our gut health and microbiome content. So if you haven't already and you're enjoying it, please give us a 5-star review because it helps us promote the podcast and push us up the algorithms. This is Inside Matters. My name is Dr. James McElroy. I hope you enjoy it. How did you get into the field of the microbiome?
Well, I guess it was a little bit just chance in a way. So when I finished my PhD in Dundee, I wanted to come back to Aberdeen, and there was a research project with Professor Harry Flint on antibiotics in rumen bacteria. So I came back to Aberdeen, did the project on rumen bacteria and antibiotic resistance, really enjoyed it, did another one, and then after that I got a job at the Rowett, a permanent job on the human gut microbiome.
And for the listener, when we talk about rumen, what are we talking about?
Okay, so the rumen is the sort of stomach, if you want, of ruminant animals. So cows, sheep, goats have got a rumen, and they have a very particular microbiota in there. It's got protozoa in it, which humans don't have protozoa. And then it's got quite a lot of fungi. We've got a few fungi, but not too many. And it's got bacteria and archaea, both of which are also resident in the human.
Right.
So it was quite a good transition using my background in rumen microbiology, transferring across to the human microbiology field.
With regards to the microorganisms in the rumen microbiota, is it the case that there's a difference between the human and the rumen because the rumen microbiota perform different functions and they're important for the animals?
Yeah, so obviously ruminants eat a very different diet from us. So their diet's predominantly fibre-based, so they eat a lot of grass, cereals, depending on what the farmer's feeding them, obviously.
Antibiotics.
And sometimes, not always, I wouldn't like to say that. But yeah, so the rumen microbiota in generally really is essential in order to provide the ruminant with energy, nitrogen, amino acids, all the components that the actual animal needs to grow and thrive. Whereas in the human, the microbiota, we should eat more fibre than we do, but that's not the main part of our diet.
Right.
So our microbiota is different, yeah.
And we're going to talk about fibre later in the episode,
Okay.
but back to the project you did with Prof. Flint. What was known about antimicrobials and the rumen microbiota at the time, and what were you trying to investigate?
I guess, well, at the time, it was known that the more antibiotics you used, the higher the level of resistance amongst the microbiota in that animal. So that was known. But what wasn't known was exactly how the resistance genes that enable the bacteria to be able to grow in the presence of the antibiotic. So a bacterium would have a specific piece of DNA that enables it to grow in the presence of an antibiotic, and that's a resistance gene. And it wasn't clear how those resistance genes spread between different bacteria and whether that spread became greater, happened more frequently if you gave antibiotics as well.
Now back to the antimicrobials.
Mm-hmm.
I think a lot of people listening to this podcast will be familiar with antibiotic resistance as a concept. And there are bacteria that develop these resistance mechanisms so that the antibiotics don't get to them.
Mm-hmm.
And I think intuitively it will make sense to people that when you give a population antibiotics, some die and some survive, and then the ones that survive replicate.
Yep.
But what you're talking about, I think, is also something slightly different, which is that those genes can be passed on from bacteria to bacteria.
So I guess the concept is exactly the same as when a child inherits blonde hair from parents. Okay? So it's the same idea. It's the progeny of the bacteria can gain antibiotic resistant genes from the parents, but then when in bacteria as well, they have very specific gene transfer mechanisms that allows two bacteria that meet each other or come together can actually swap DNA between them if the DNA has some extra particular features about it. So it has to have like transfer genes, and the bacteria have to be able to what we call conjugate, which is basically come together and form a bridge between them, and then the DNA moves across that bridge into the other bacterium, and that's a way of spreading the resistance between two different bacteria that otherwise may not have anything in common, if you want.
Right.
And that was one of the things in that project. I was trying to see how unrelated bacteria could be and actually still transfer DNA between them.
What are the determinants for the transfer? Like, there has to be an acceptance of them and there has to be a push. So what happens there?
So sometimes there is a push. So the antibiotic selective pressure in itself would be a push, because bacterium B can't survive unless it has a gene to give it resistance.
Got it.
And it knows bacterium A's got that gene.
How does it know?
Well, because bacterium A is going to overtake the population, so it senses more and more of bacterium A is taking over. Yeah, so bacterium have, we call it communication systems between bacteria, and different bacteria communicate with each other in different ways, but one of the ways is that they release things into the environment that other bacteria can then pick up on. And it's like a sort of pheromone sensing, if you want. Once the population gets big enough, then bacteria do different things based on that knowledge.
Wow.
So gene transfer is one of them that they can do. Probably
a massive step too far, but is there any school of thought within the field about bacteria being conscious?
I don't know. I wouldn't comment on that, but I do when I'm telling students about it. They're cleverer than we sort of give them credit for, if you want, because of the fact that whenever we develop a new antimicrobial or an antibiotic, they will overcome it. I mean, that's just a given.
Yeah.
It's just how long does it take? That's the difference.
Got it. So they can sense there's a lot more of these ones surviving. I maybe want a bit of that. Mm-hmm. And the ones that are surviving are going, we're surviving, we maybe want to give the others a bit of that, essentially.
Yeah, that's true. So sometimes it might be to their benefit to allow that other bacterium to survive as well, because bacteria also interact with each other in lots of different ways. Sometimes one bacterium can't survive very well in an environment unless there's something in the environment that's produced by its friend. So it's in both their interest to survive together. So they are cleverer than we give them credit for, really. Yeah,
super, super clever. It's sounding like there's a heck of a lot of complexity within these ecosystems.
Yeah.
And how do we model the communications, given that there must be so many happening all the time?
What you mean in the lab? How do we model it? We can't really model them all. We can do our best to recreate microbial ecosystems, but usually when we recreate them, there is something missing, or we add in something rather more synthetic to mimic something that would be produced naturally otherwise.
Right.
There are different ways that we try to get around it, but I wouldn't like to say that we managed to model the whole ecosystem terribly well.
Right. Now, on the antimicrobial side, did your project really open your eyes to sort of what was coming over the next few years and longer around we've potentially got a very big problem here?
If I'm perfectly honest, probably not. I mean, I don't think at that time, so that was in the 90s, I don't think we really thought we would be facing this almost existential thing about antimicrobial resistance, because, I mean, the current thinking is, as you probably know, by 2050, antimicrobial resistance will be responsible for more deaths in a year than cancer. I mean, that's a big thing, and we're causing that problem. And I think, I didn't realise. I mean, now many scientists know this is an issue now, and there are lots of things that are going on to try and not get to that problem point. But yeah, I wouldn't have seen that coming, no.
By 2050, many people.
Well, that's the last reading I read, yeah.
That's really scary, isn't it? And
well, it is, because cancer, we always think of cancer as being a big killer.
Yeah.
And but I suppose the flip side of that is that cancer isn't the killer it used to be. So the two things are balancing each other out a little bit. So people recover from cancer. So the number of deaths due to cancer are decreasing. But there is definitely this problem of antimicrobial resistance.
And you can map really nicely the amount of money invested in cancer diagnostics, therapeutics, prophylactics, all that research and development and the screening, versus what we do on the antimicrobial side. And I don't know what the delta is, but it's massive. So, imagine if the same level of effort went into the AMR side as it did to cancer. I think we would see some pretty rapid innovation.
Yeah, that's a very good point. There certainly hasn't been the research on alternatives to antimicrobials the problem, as I mentioned before, because bacteria overcome new antimicrobials and become resistant to them, there's not a big drive for companies to develop new antimicrobials either, because companies are not going to spend millions developing something that they can't sell in 5 years.
Indeed. And also the other thing to add to that is, if you develop a new cancer therapeutic and it's better from an efficacy perspective, it jumps right to the front of the queue. And therefore you immediately leapfrog what then becomes the second best treatment. So your sales go. On the antimicrobial side, however, it's typically saved because you don't want to develop resistance against it, so nobody buys it.
Yeah.
Which is one of the main challenges. And we don't have a solution for it, unfortunately.
No, that's very true. The best antimicrobials are kept as antimicrobials of last resort.
Yeah.
And we have to do that. We can't not do that, because those are essential for those diseases that have got multiple resistances that can't be treated by any of the more generic ones that are widely used. So you're right, that is a big issue for the industry development.
And are there bugs out there right now in the world, not trying to alarm the listener, but are there bugs out there right now that are resistant to everything? Have we seen that?
No, there are still antibiotics of last resort that work on what we call the superbugs that are resistant to, I mean, most people have heard of multiply resistant Staphylococcus aureus.
Yes.
Some enterobacteria that are resistant to lots of antibiotics, vancomycin resistant enterobacteria, for example. Yep. We do have antibiotics that we can use to treat them at the moment. But that's why it's really important to keep those ones for critical times when they are needed. Right.
And what about nature's antimicrobials? So there are a few of those, aren't there? Is that something we could potentially exploit?
Well, that's actually something I'm working on at the moment.
Good! I might have known that.
So,
tell us.
Well, it's one of the, bacteria also control each other in a lot of ways. So something that you can possibly do is use one bacterium to fight against another one in very targeted ways. And there's also other groups in the world that are harvesting molecules from certain bacteria that only, they're very specific. So an antimicrobial, that's one of the problems with antimicrobials, for example, is that they're quite broad spectrum, which basically means they kill lots of different bacteria. Whereas some of the bacterial-derived products that are being looked at the moment can just kill one target bacterium. And that's that makes them very effective, because they don't have there's no collateral damage. That's also something I didn't mention yet. So the collateral damage of an antimicrobial is something else. But these targeted antimicrobials that kill, say, Clostridioides difficile, they are being developed and tested out in different labs across the world.
So to break it down for the listener, the classical antimicrobial is like a small molecule, which has an effect on pathways, and sometimes it's a cell wall and these kind of things. And typically the mechanism would have an impact on many different types of bacteria, and they can be classified based on various different levels. Like we use gram staining, for example. We can talk about species, genus, and that kind of thing. So try and help the listener understand how broad some of them can be and why that's potentially a problem.
Yeah.
And you talked about collateral damage, which you should pick up on as well.
So if we think about bacteria, we really can describe bacteria in two very generic ways. We have gram-positive bacteria and gram-negative bacteria, and that's based on different types of cell wall. And that's the single difference between those two types of bacteria, and all bacteria will either be gram-positive or gram-negative. And some of the antimicrobials affect gram-negative bacteria generically. So any gram-negative bacterium will be affected if you use one of those broad-spectrum antimicrobials. And that then, in terms of collateral damage, if we talk, and equally you have other antimicrobials that will only affect gram-positive bacteria. So in the collateral damage part, if you have an antimicrobial that affects gram-positive bacteria, and if you think the way that most people take most antimicrobials is by swallowing a tablet.
Yes.
So that tablet goes straight down into the stomach, through to the small intestine, into large intestine. The large intestine is the biggest microbial population in our body. Many of those bacteria are gram-positive bacteria. So that antimicrobial that you're taking for your bacterial ear infection is circulating around your body in your bloodstream, but it's going to your large intestine and also affecting the bacteria in there.
Got it. And that could be a problem, right?
It can be a problem. It can normally, for the general person who's generally healthy, taking antimicrobials for a sort of routinely prescribed course of antimicrobials, 5 days, 10 days, whatever it is, it can be a sort of short-term problem. So it will have an effect on your microbiota for the time you're taking the antimicrobial, but after you come off it, your microbiota tends to come back to pretty much what it was before.
How long does that take typically, or does it sort of, is that one of those?
Probably varies, yeah, it probably varies. I mean, with it, people have done studies a month later and most things are back to normal. It is most things are back to normal.
Is there anything people can do to try and accelerate that reconstitution, if you like, or the enhancement or the restoration of their microbiome post antibiotics? So
I'm sure you're asking that question, trying to lead into probiotics. So I would say that the evidence for something to actually enhance the recovery of what was there before is not really a lot of it. Okay, it just needs time.
Yeah.
Basically. However, what you can do is prevent any sort of bad things happening during that period of time when your own microbiota has been affected by that antibiotic. So if we try and think of an analogy that people can sort of visualise, if I think of like say if you've got the Amazonian rainforest, so you cut down the rainforest, you're destroying everything that's there. That's what the antibiotic can potentially be doing in your large intestine. It's destroying everything that's there. It might leave a few things and they can start to grow again.
Yep.
However, if you go back to the rainforest, if you replant the rainforest with 1 or 2 species of tree, you still have a forest, but it looks very different from what you had before. Whereas and sometimes some of those things that might grow are not very good for the forest regeneration. So I don't know if you say you've got lots of ferns growing instead of trees in that forest. You might not want those ferns to grow. And in the intestinal ecosystem, what can happen is that bacteria that are normally kept under control by the other bacteria that live there can actually expand their populations, and that could become a problem. And in order to prevent that, you can take a probiotic, and that can prevent that from happening. And there is evidence that if you take probiotics alongside antibiotics, you can stop getting diarrhea, for example, or things like that.
And in some countries, it's just standard that they prescribe a probiotic alongside a antibiotic.
Yeah, I think that's...
Italy, for example, they do a lot of that.
Some countries are a bit more open to the idea of probiotics, maybe, than we are in the UK.
Right.
We can't even use the word.
No. I mean...
Well, in the EU, you can't use the word a probiotic, but everybody knows what a probiotic is. Well, many people know what a probiotic is, even though none of the marketing can actually use that single word on it.
Because in the eyes of the regulator, they've not demonstrated the health benefit.
Yeah, so probiotic, the sort of Latin translation, in my understanding, is that means for life. So
that's
by
lovely.
using the word, you are saying that this product improves life.
Right.
And that's a very hard claim to prove. So the legislators have said, well, you haven't proven that your bacterial product, probiotic, improves life, so you can't say it's a probiotic.
I did kind of lead us into probiotics, but I want to spend more time talking about that and all the work that you're doing. I'd like to bring us back to this collateral damage concept. Right. I think it's kind of fascinating. What could the downstream negative consequences be of collateral damage? So, someone's got an ear infection or someone has a throat issue and they run to the doctor and say, I need this, even though it might be viral.
Well, that's a big problem.
Might be viral.
Yeah.
And look, I just need this.
Do you know why that's a problem?
Am I being interviewed now on my own podcast? I do know why.
Yeah.
And I might get the exact percentages wrong. However, I think something like 40% of classical upper respiratory tract infections are viral, might even be more. And our ability to prognosticate, if you like, or to diagnose whether it's bacterial is very poor, based on the fact that we don't have real-time live diagnostic capability. It has to be a swab and it's sent to lab, and before you know it, you're actually well again. There are certain criteria. One's called the Centaur criteria, if I've got it right. Really taking me back now to my medical days. And you sort of feel, have they got any lymphadenopathy in their neck and cheeks? Do they have pussy tonsils? Do they have a fever? I might be getting this wrong. I'm sort of surprising myself that this is coming out because I thought I'd forgotten all of the medical student stuff. So well done to the Aberdeen University Medical School lecturers. But my understanding is a lot of them are viral, and your antibiotics will do no good for your viral infection.
Yeah. So your answer was much more complicated than mine would have been. My one would have basically been antibiotics don't work against viruses. Okay. Antivirals.
I thought you were really testing me because I can see those notes, and I thought, I'm going to flip it round on James today. Oh man, that was funny.
So no, I mean, an antiviral is required for a virus infection, but you're quite right. We don't know whether the infections are caused by bacteria or virus, and usually, or as you say, a lot of them are viral, and an antibiotic is never going to work. Right. Not even if you took it for 100 years.
No. and it's just, we talk about the collateral damage, are there potentially negative consequences associated with
this? So during that time when your own microbiota has been affected by the antibiotics, as I say, you can get blooms of certain bacteria, and if those bacteria cause disease in themselves, then that can have that negative effect. One of the classic examples of that is people in intensive care who are getting vancomycin treatment, and that destroys many of their gut microbiota. And this particular bacterium called Clostridioides difficile, or C. difficile, really can increase in number very rapidly in those people. And because that bacterium, unchecked, can produce toxins that interact with the cells lining the gut, it just causes a huge problem and can actually lead to the death of the person rather than whatever they were in intensive care for in the first place.
Now, vancomycin is very interesting. You may not know this, but you may also know it. Many of the microbiome companies who are developing what I'll call bugs as drugs, next-gen probiotics, these defined consortia and single strains, give a course of vancomycin before they give their probiotics or LBPs to try and promote what they call engraftment. Now, I've always been a big critic and said, look, come on, you're giving these people vancomycin, knowing all the risks, just so you can promote the engraftment of your drug. I mean, I'm now going on a rant, but I'll just do it. Let me indulge myself. Surely we can't be aspiring to giving our patients vancomycin long term to promote engraftment, quote unquote. There has to be a better way of doing that. And I personally would say, no, I don't want your vancomycin. Thank you very much.
And of course, why they're doing that is to clear the way
for
their engraftment to work.
That's right, yeah.
So they're deliberately decimating the population.
Correct. And what are your thoughts as a microbiome researcher and expert on that? I
mean, I think if the person has had vancomycin to treat a disease in the first place, then that's a different thing. That's happened because the vancomycin has been applied for a legitimate reason. But to use vancomycin purely to clear the playing field of the opposition and allow the new players to come in and win just seems bizarre and wrong somehow.
Yeah. Well, it's happening.
Well,
that's what they're doing. So that's what the future looks like at the moment.
Well, hopefully they've got the evidence that actually the reason why they're doing it are valid.
And to be fair, they are doing well-designed studies to actually test that specifically.
Okay. Well, we'll wait and see.
So there's one vancomycin arm, one no vancomycin arm. So they are testing it, to be fair. They're not just saying we want to do this. But the originator, if you like, I believe, and of course I don't work for them, and we have a different approach at EBX, but it came through trying to convince people that there is a mechanism of action and quote-unquote engraftment is part of that story. And the animal model evidence is really quite convincing in terms of if you give vancomycin, then you create the niche and you show engraftment. So it's part of sewing that story together. And there have been some high-profile failures as well. And some people say, well, they failed because they didn't clear the field.
Well, I think that part is probably true. I think microbial engraftment works when there is an empty playing field, because all of those bacteria in the large intestine are living on the food that people eat, and they have their own niche that they occupy, and that tends to, in a healthy adult, that's stable for years. So if somebody else is in your niche, you're never going to be able to take over that niche. So that's why microbial engraftment without that clearing of the playing field is a very difficult thing to do. But it doesn't necessarily mean it's the right thing to do.
Well, I'll give maybe some food for thought for yourself and the listeners. So in other fields of medicine, clearing the field does happen. So in these stem cell transplants, for example, they get radioablative therapy. They get massive total body irradiation, and they get chemotherapy to suppress all the blood cells. And then you get the stem cell transplant after to sort of repopulate. So they might be saying, well, this happens elsewhere and therefore it should happen here. But there's one other way of the clearing the field. It's the laxatives, what they call the bowel lavage. And that obviously has limitations as well for the patients because they're running to the loo.
You would think that was slightly less, long-term detrimental.
Yeah.
Because everybody has episodes of diarrhea in their life, and their microbiota recovers back to what it was before. So
Yes. The EBX view, the Enterobiotics view, is, well, because our product is so diverse, we have a higher probability of our product finding niches within the gut. Whereas if we had a small group for whatever reason in that particular patient, they might just get booted out. That's the view we've taken. More, we call it shots on goal.
Okay.
And we're not doing vancomycin or bowel lavage as part of the value proposition for the patient.
But yeah, and you're giving it to patients who have a disturbed microbiota.
Yes.
So
invariably.
Yeah. So you're right. If you've got, there may well be a niche in there that's able to be occupied by some of the incoming bacteria.
Yes, we hope so. We're testing that in the clinical study. Now, on collateral damage, we've talked about some of the potential negative effects. One thing I'd just like to pull out, which you mentioned, which I think is really interesting, you talked about the microbiome being stable for years. Does the microbiome change from when people are born and as they get older, and when does it reach a kind of steady state, if you like?
Yeah, so the infant microbiota is very different from the any post-weaning microbiota, let's say, because as I mentioned earlier, your large intestinal microbiota, we are speaking about the large intestinal microbiota predominantly. We have microbial all over our body, but the large intestinal microbiota relies on our diet to feed it. And an infant's main diet is milk, which is very different from an adult diet.
So just, like I said, why is the large bowel the most important? Is it?
Well, somebody working on the small intestine would argue that that's not true. But it's most important to me because I work on the large intestine. But it's also got the largest population.
Yes.
So it is the most populated, microbially populated site in the body. It's about 10 to the 11 microbes per mil of content, which is more people than live in the world, actually.
It's a lot of bugs.
It's a lot of bugs.
There's a lot of bugs there.
And that's in 1 mil, and everybody's bowel, unless you've just been to the toilet, contains about 200 mils of content. So you can multiply that 10 to 11 by 200 to get the actual number of microbes in the adult large intestine.
That's really a lot of bugs.
It's a lot of bugs.
We are, residents. They're residents within us.
They are, well, we're basically their mode of transport around the world, if you want. And there's more microbial cells in or on us than there are human cells for every person.
They might all be laughing with, this is great, we've got this home and we get fed every day, whether,
Yeah, well, it's true. It is an interesting...
Maybe they're controlling us.
Symbiosis.
Could
do you really want to go in?
A little bit, yeah, a little bit. I do enjoy that kind of off-piste comment, I'm not going to lie.
I'm not sure about controlling us, but they certainly send signals to our brains as well. So there are, if that's control, then...
maybe, yeah. What kind of signals?
So when these microbes digest our food, they produce what we call fermentation products, because fermentation is the process by which they degrade our food. And the products that come out of that fermentation include, one of the specific products are short-chain fatty acids. And these short-chain fatty acids get absorbed into the bloodstream and transported around the body. And one of them in particular, we now know is really important in brain signalling, for example, and helps us to feel full for longer. So it's important in satiety signalling.
Are any of the short-chain fatty acids more important than others, or is it kind of they're all important? So
there's 3 main short-chain fatty acids that are important around the body: butyrate, propionate and acetate. And they all have different functions, and they all interact with what we call receptors that line the gut wall in different ways, and different short-chain fatty acids are preferred by different receptors.
Right.
So for example, one that I do quite a lot of work on is butyrate, and that one is used by energy for the cells that line the gut. So most butyrate's actually used up in the gut itself, because all the cells lining the gut, that's their preferred energy source. And lack of butyrate means that those cells don't turn over properly, and then that can cause issues in the health of the large intestine.
Right.
Propionate gets transported to the liver and is also one of these brain signalling molecules. It interacts with the gut receptor that produces a gut hormone that then send signals to the brain.
And when you say it goes to liver, like what happens in the liver? Is it part of this gut-liver axis that we talk about?
Yeah, so it's a monitoring thing for gluconeogenesis in the liver. And how that actually works, I'm not 100% sure.
Yeah, there'll be a lot of
it's complicated.
pathways and arrows and things like that. Now, this does bring me quite nicely into one of the topics which I wanted to discuss, which was fibre, and kind of what it is and why is it important. You said earlier on we don't get enough of it.
Yeah, most people don't.
Most people don't, yeah.
Most people living in this country anyway, and most people living in the Western societies don't get enough fibre. Right. Yeah.
Right.
So I think the recommended intake is 30 grams a day, and most people get less than 20. So it's quite it's a big deal.
Big picture, what is it? And
so, I mean, the easiest way of thinking about fibre is it's like a big component of plant-based foods. Plant-based foods that we can eat. So fruits, vegetables, cereal products, all these sort of things contain a lot of fibre. As long as they're not refined, it's not refined out of them. So I mean, like white wheat flour, for example, contains very little fibre, whereas whole wheat flour contains a lot of fibre. So that's important. Is you have to take in the whole grain is the part of the cereal that you really need to take in.
Minimally processed, essentially.
Minimally processed. Well, minimally processed for any food is really the best way to go if you can.
Right.
Yeah.
That's been a recurring theme on this podcast. I'm so happy you said it. Yeah, that's one way of describing it is this made, and this is, caveating this, I'm going to say something which relates to God, but if it's made by God, then it's probably good. If it's made by man, it probably isn't. So can you pull it out the ground, or does it have to go through some manufacturing process or whatever, in terms of the minimally versus maximally processed?
Well, that would be going to refined foods are certainly maximally processed, and you definitely don't want to eat them.
Yeah.
But some processing can actually help us. So some processing can improve the sort of availability of the fibre. So if we ate grass, we would not really be able to digest grass, and that goes back to our conversation we had about the ruminants at the beginning. Whereas if somebody's taken grass and extracting a specific thing from that grass, then we could potentially, grass is probably a bad example, but we could potentially digest something then. So some processing might actually be okay. But yeah, you are right that the more processing that's involved in the food, the worse it is for you generally.
Got it. And I was going to do some sort of silly thought experiment around what if we did a transfer of rumen microbiota into you and eat grass, but I'm not even going to go there because we're on a good roll here.
I don't think we should.
No, we're on a good roll here, and I've sort of...
There are other differences as well that would be important. You can't just turn me into a ruminant by doing a transfer like that.
Like, you've only got one stomach and some animals have more than one stomach.
Precisely.
Yeah. So, on fibre then, why is it important for the microbiota? It's because they eat it, essentially.
Yeah, so they're basically reliant on our diet for anything that they eat. And this comes back to the highly refined diet as well. So a highly refined diet tends to be really high fat and high sugar. And sugar is absorbed in our upper intestine. It doesn't make it as far as the large intestine, where most of the bacteria live. So they're not getting any benefit from your Mars bar that you had before you came in here.
Oh, don't worry, I wouldn't. I would never. Not on a Monday, anyways.
Friday food.
I can be tempted into. I mean, you don't even ask, but cheesecake is a real weakness.
Everybody can have treats, though.
It's real weakness. Yeah. It's very difficult. If I see it on the menu, I just go, "Oh no."
Yeah, but a little of what you like doesn't do any harm. But
Agreed.
But going back to the sort of healthy diet, fibre sort of thing, sugar gets absorbed in this upper gut, if you want, whereas the sort of plant-based fibre products reach the large intestine, and they're therefore available for the bacteria that live there. And that's why if you don't eat enough fibre, you're not feeding your gut bacteria properly. And it ends up that they have to feed on whatever is going in there, which might be protein or fat, and that tends to be converted by the wrong type of bacteria into the wrong type of products that can actually cause damage in the intestinal ecosystem.
And we're specifically talking about the metabolites that they produce maybe having a damaging effect on the body. Like, could you give an example? Like TMAO, would that be one?
So TMAO is not produced by gut bacteria. So gut bacteria produce trimethylamine, and trimethylamine is transported to the liver, where it's converted into TMAO, trimethylamine oxide, which is then implicated in heart disease, for example. So that is a bad product. But I was more thinking of more general, if you digest protein, if too much protein reaches the large intestine, one of the byproducts of protein degradation by gut microbiota is ammonia.
Yep.
So even you don't really think ammonia is a very great thing, and it's not,
because
bleach,
it's right.
Well, it's in bleach, but it also is quite alkaline, and the pH of the large intestine should be slightly acidic. That's the optimal pH. So the more alkaline it gets, the worse it is. And another product of protein digestion is hydrogen sulfide.
Yep.
Which is really bad for the lining of the gut. It's a really inflammatory molecule, and so it can cause lots of inflammation, which is never going to be a good thing.
So for protein's kind of interesting though, because I think for maintaining your muscle,
we need
longevity,
some.
we need some.
Yeah,
but
we do need some, but I think most people in the West tend to have too much protein in their diet,
and not enough.
And there's a limit to how much your body can actually use for muscle regeneration, all these sort of things, and anything that's left over is going to reach that large intestine,
or get peed out.
Yeah,
as an amino acid, or urea.
That's when it's been degraded. Yes, it's been broken down. But in your balanced diet, if you have more fibre replacing some of that excess protein, then you're going to be feeding your gut bacteria with better stuff rather than the protein.
And is there any fibre that's better than another fibre? Because it promotes specific types of bacteria?
Yeah, so that's an interesting question. I would say that every type of fibre is important. So in terms of that microbiota, those 10 to the 11 bacteria, you don't want them all to be the same thing. You want them to be a very different, diverse population. And all those different types of bacteria will like different types of fibre. They'll have their own preferred food. Like you said, you would not go past cheesecake. I wouldn't go past a chocolate brownie. So those bacteria are the same, right? They have their different preferred foods. So you should really be taking in a lot of diverse types of fibre.
If you come on again, I'll have brownies, don't worry.
Well, there you go.
A slice of cheesecake.
Yeah. So you really need lots of different types of fibre. And I was listening recently to somebody speaking about this, and they said that everybody should try and eat 30 different plant-based foods a week.
Wow.
And initially I thought, well, that's an awful lot. And that day, it was in the summer, so it was easier. But I was making a salad for dinner, and I got 10 different plant-based foods in the salad. And that was a great start for the week.
That was a great start, yeah, I smashed it.
Yeah, so, but it's quite a good thing to think about, because it does make you actually think, instead of having an apple again, well, maybe I should have a pear, or maybe I should have an orange, or have something different.
That's really interesting. Well, before we started recording, we talked about your lunch, and I won't tell everyone what you had. But
wasn't that bad?
No, it was good. I thought it was good, very nutritious. I over the years, having got more and more interested in the microbiome and the importance of diet for gut health and microbiome and overall health, I have probably hit that 30. I reckon. Every day I'm having 5 or 10 different types of fruit.
Yeah.
Orange, blueberries, raspberries, strawberries, bananas, apple, mango, sometimes pear, sometimes peach. And
it's a lot of fruit.
It is a lot of fruit, but we have these huge fruit baskets in the office.
Well, that's a good thing. Yeah, every workplace should do that.
Yeah, and the fresh pressed green juices. And people do go for it. And if there's an element of, if it's there. And
it's
now,
true.
I also believe now, you might say this is ridiculous, I kind of crave that a little bit. So if I'm, a lot of these networking events, particularly in the US, but also in the UK, is breakfast networking events. And you turn up and you see a table like this, and it's just full of pastry and orange juice and jam, peanut butter, and you just think, where is the fruit here?
Yeah. You're right.
You know, or we have a bacon butty. Now, don't get me wrong, no harm to the bacon butty as a Scottish staple with an iron brew, but, you know,
sometimes.
Yeah, not every day, right?
No, you're right. And I think it's just as well that you're not at those networking events every morning and you're having your porridge on the other mornings in between times.
Yeah.
But I think it's an interesting concept because you're right. You do start, after you start getting used to having a diet that's healthier, if you want, that's got more fibre in it and more fruit, and I would say vegetables. You probably need to get your vegetable intake up there. Oh, you haven't got to that bit yet.
I mean, okay. I could if you want, but I'm not there yet. But thank you. No, any advice is appreciated. Sorry, this is an open forum. Any advice is appreciated.
But you're right, because I would find that as well if I go, and my husband actually, even like if he goes away, he does like hill running. I'm sure you don't mind me mentioning him here. He does like a lot of hill running events, and you're on dried, reconstituted food because you're carrying everything with you, and every gram of weight matters. Yeah. And he comes home from that and straight to the fruit bowl and munching on the fruit because he feels he's been deprived all weekend really or something.
Does he do some crazy things?
Yeah, not as crazy as some people. It's not like these long distance mega marathons.
Just fell running,
mountain marathons over the weekend, but it's a marathon over 2 days rather than.
That's cool.
Yeah. So.
Did you do one recently?
He did. There was one this weekend actually up near Braemar that got cut short because of our lovely weather this last weekend.
He was at that one, yeah. That's cool. A few weeks ago, I was at one in Kinlochleven.
Okay.
Ring of Steel Sky Race. And there was also a marathon that day as well.
It would have been better weather then.
Honestly, perfect.
Wow.
It was like bluebird.
Yeah. Excellent.
I couldn't believe it.
Yeah, that's nice.
The time beforehand was in Glenshee, and it was dreadful. But this weekend past was just...
Yeah. Well, I would just recommend that nobody ever does one at the same time as my husband because it always rains when he's doing it.
It's a bad omen.
Word to the war, yeah.
Yeah, the craving thing, I think it's a real thing. Maybe could it be, and again, I'm not trying to get you to say bacteria are conscious, but could it be something like, you know, the microbes change in your gut and they're expecting this, you know, array of fibre, and if they don't get it, they produce signals saying, where's my dinner? Or my breakfast.
Maybe, I don't know.
Do they sleep? Do bacteria sleep?
That's a good question. No, they don't. Well, E. coli's got a 20-minute turnover cycle if it's grown in optimal conditions. So E. coli is one particularly fast-growing bacterium, and if it's got a 20-minute turnover cycle, it can't really have a lot of time for sleeping in there.
No. Okay. Just thought I'd ask.
So no, I don't know if it's the bacteria that crave it or if it's our body that craves the products that the bacteria make. That probably makes more sense to me.
Interesting.
That it's the bacterial metabolites that we're used to detecting them going around our body. So
that's really interesting. Never heard it described like that before.
There probably is a craving somehow, but yeah.
That's really, really fascinating. Right. So if someone wants to increase their fibre intake, eat more fruit and veg? Is that the best advice?
Well, fruit and veg, but also cereal fibre. So cereal fibre is really important as well, and beans and pulses. It's not just fruit and veg. So I know that they've done surveys in Scotland. Well, everybody has heard the 5 a day message for fruit and vegetable intake. And they do surveys in lots of places in Scotland. You ask everybody in the street, how many portions of fruit and veg should you eat a day? 5. How many do you eat a day? And it's not often 5. And I think that's because some people just don't really like fruit and veg. So I think the important message is it doesn't have to be fruit and vegetables. Okay. If you're having whole grain cereals or whole grain toast, something that you like, just make it a high fibre type of that something. And then you're actually helping out your gut bacteria as well. So I think it's really difficult if you really hate a certain type of food, it doesn't matter how many people tell you you've got to eat it, you're not going to like it any better. So alternatives are important. And beans, pulses, cereals also are really important fibre-containing foods that can, I wouldn't say replace fruit and veg, because I would say take them as well as your fruit and veg. But if you really don't like it, then that could be a useful alternative.
Thank you for that. That's some really excellent advice. I'd like to ask you what I think is hopefully an easy question. What is a healthy microbiome?
You knew that wasn't an easy question. So there is no single defined answer to that question, as you well know, James. So there you go. That's my answer.
No,
there is no
come
answer.
on, please. We need your thoughts on the matter. And I know I'm kind of winding up a little bit, but I think you're enjoying that. So
the healthy microbiome is a continuum, really. So we started speaking about the small children pre-weaning have a different microbiome from an adult. It's not unhealthy, it's different because it's adapted to their diet. And then an adult has a different microbiome. Now, you and I have different foods that we eat. I don't have quite as much fruit as you do per day, for example. And there's 3 people in this room, we all have a very different microbiome. And I wouldn't presume to say that any single one of us had an unhealthy microbiome. So there's different states of microbiome can all be defined as healthy. I think what's easier to maybe make a comment about is what makes an unhealthy microbiome. So that's slightly easier. So we know full well that an abundance of certain types of bacteria, specifically proteobacteria, is really not good for health, because they're of many types of those bacteria are pro-inflammatory. They also tend to be more able to grow in oxygen. So if there's an abundance of them in your microbiome, that tends to be a sign that there's too much oxygen in the large intestine. So I don't know if we mentioned. So the large intestine as an environment for bacteria is really anaerobic. So there's very little oxygen there normally. There is some, because obviously our blood contains oxygen and blood flows all around the body, including around the large intestine. So there is some oxygen near the surface of the large intestine, but there are many different parts of it are very anaerobic, like they don't have oxygen. And the vast majority of the bacteria that live in there are also obligately anaerobic, and that means they can't live in oxygen. Whereas these proteobacteria do live in oxygen. So too many of them is a sign that there's more oxygen there, which is a sign that there might be inflammation.
Yep.
Because there's more oxygen exchange from the blood's more close to the surface and everything. So that's one thing that certainly would be almost a definition of an unhealthy microbiome.
Can these proteobacteria be inflammatory themselves rather than being, potentially present as a virtue of inflammation?
Yeah, they're pro-inflammatory as well. So you start to get if you have an inflamed large intestine, it almost starts to be a vicious circle, because your inflammation increases the amount of oxygen. The amount of oxygen increases the amount of proteobacteria. Right. The amount of proteobacteria interact with the cell's lining to make inflammation. And then the inflammation increases the oxygen, and then it just goes on and on and on. Unless you actually interrupt that cycle, then you're going to have some problems.
So the educated listener might immediately jump to the conclusion of, well, how do we get rid of the pro-inflammatory proteobacteria?
Well,
is there a way of doing it?
That's like a big question. Most people's healthy gut, they are kept at bay by the fact that there's not so much oxygen anyway, and there's other bacteria there to occupy those niches. And if you have enough fibre in your diet, then you're making sure those other bacteria keep at a level that's high enough to not allow those proteobacteria to take over. And so when they do take over, it's probably a sign that the other bacteria have reduced in number, whether it's because of the oxygen or the inflammation or whatever other impact, or antibiotics that have been consumed or something. How do you get rid of the proteobacteria? I think outcompeting them by trying to boost your other bacteria is probably the easiest way to do it.
Yep. When we say out competing, we're meaning we're sort of stimulating the growth. So if you think back to the rainforest, we're trying to get them to grow.
Yeah.
And is that the fibre coming into play again?
Yeah, so I would say the fibre would come into play. So if you provide a food for the bacteria that the sort of good bacteria, if you want, by increasing your fibre intake, and the proteobacteria might not be, often aren't, in fact, as good at competing for that food.
Yep.
So they wouldn't get so much food, so they'll actually start to die out naturally because they can't turn over because they're not getting any energy to grow. So that'd be one way to try to address that dysbiosis, if you want.
That makes sense. What happens to bacteria when they die in the intestinal tract? Do they just get...
Yeah, they'll just get washed out in your poo and you wash down the toilet.
They're dead.
Mostly.
Okay. Mm-hmm. Now, bringing us back to the precise means of destroying bacteria. So we talked about the antimicrobials, the classical antimicrobials. Could you tell us about some of the things you're working on with regards to AMR?
As alternatives to AMR, you mean?
Yes.
So I think we've sort of mentioned the fact that bacteria interact with each other. So often that's beneficial interactions that both bacteria or bacterium A produces a product that helps bacterium B survive. But what you can also get is that bacterium A might produce something that inhibits the growth of bacterium B. Mm-hmm. And one of a very simple example of that is actually the short-chain fatty acid production that I mentioned before. So there's short-chain fatty acid production through fibre fermentation by your gut microbiota produces, well, the short-chain fatty acids themselves are acidic. So then you get a change in the pH in the large intestine. And that slight lowering of the pH in itself inhibits the growth of pathogenic bacteria. So the likes of your E. coli doesn't grow terribly well at pH 5.5, for example, whereas it might grow better at pH 6.5. So the more you can keep the pH of your large intestine down, the less of that can grow.
Interesting.
So that's a sort of generic effect, if you want. But the other thing that some bacteria can produce is they produce very specific products that are they're called bacteriocins, but basically they're like often a small peptide molecule, so a small protein that actually acts against a very specific other bacterium. And the bacteria do that because they don't want to compete with that bacterium. So those two bacteria might often live in the same habitat. They might fight for the same resources or something. So the bacterium A produces this thing that tries to keep bacterium B in check because it wants to use that resource. And we can capitalise on that by identifying those types of tiny protein molecules that are active against, say, the highly pathogenic bacteria that we want to destroy.
Yes.
And if we can manage to find those molecules and purify them or something, then we can use them as alternatives to antibiotics because they're so specific.
Do the classical resistance mechanisms exist for these bacteriocins?
Not that we know of so far. I mean, whether or not they would arise if you used them broadly and widely, I don't know. But I mean, those bacteria have lived together, I suppose, in the same niches for quite a long time, often. So it's possible that they don't arise, but I don't know. We would have to wait and see.
I don't like to use kind of war analogies, but it sounds like the antibiotics are like big bombs, whereas these are like bullets or arrows.
They're much more targeted.
More targeted.
And that makes it better because you don't have the collateral damage that we were speaking about before.
Right. How far away are we, do you think, from, a doctor saying, you've got this vancomycin-resistant enterococcus infection, and instead of giving you teicoplanin, it's probably resistant to that, instead of giving you whatever we give next, we're going to give you just this capsule or a wee shot that contains bacteriocins?
So for some bacteria, they're probably not that far away. So there's some research going on in a few different research groups around the world, actually, focused on, say, Clostridioides difficile. There's another one against a bacillus species that causes disease as well. So it's not like decades away. It's certainly in the next 5 years or so, say, maybe, for certain bacteria.
It's exciting.
We don't know for all of them, though. We don't have those products yet, and they are often very hard to identify
because they're so small, or
so some of them are like, say, 10 amino acids.
Wow!
So they're tiny molecules.
How do they work? What happens?
I don't know the mechanisms exactly, but I think they must interact with something on the cell surface of the other bacteria, and I don't know if they affect the structure of the cell wall then, and the bacteria just lies.
It's incredible that they're so simple at amino acid level, but have such a powerful effect.
Yeah, well, that's true. And some of the bacterial communication signals are about that big. That's the sort of size that you are talking, yeah. So, and they're because they're so tiny, it's really hard for us to find them. Even if you've got a gene, I don't know if when we sequence bacteria, so if you've got a whole genome of a sequence, most genes that encode proteins are like 1,000 bases or 2,000 bases long, and that encodes a protein of, say, 800 amino acids. So these things are 10 amino acids. That's 30 nucleotides. That's your signal in your massive 2 megabases of sequence. So it's really difficult to find. It's something that artificial intelligence might be able to help us with in the future.
So some people talk about this like dark matter of the microbiome, because we've profiled a lot of the bacteria, but there's still some we haven't profiled, and there's also all the things that they produce. Should we talk a little bit about what we understand and what we kind of don't understand about the microbiome? Yeah.
So in terms of culturing, we've come a long way in the last 20 years in culturing bacteria, but you're right, we can culture using the specific media. We can culture all the most abundant bacteria that are there. The less abundant ones are much harder to get a handle on, because you're looking for 1 out of 10 million maybe, and it's really difficult to get rid of that 10 million to find that one that you're looking for. So again, you need to, this sort of whole what we call metagenome sequencing. So the genome sequence of the entire thing together might actually be able to help in that respect, because you can take that whole soup of bacterial DNA sequences and then try and extract out all the individual components of that soup. So that gives you your individual bacterial sequence of those bacteria that you were never able to culture before, so you didn't know were there.
Right.
And if you look at their genome sequences, you can maybe find out specific things they can grow on, so you can design a media that then allows you to really target in a targeted way to culture those bacteria. We're not done that yet, but that's a sort of thing that could happen.
The perfect media.
Yeah, the perfect media, or it's a bespoke medium for that individual bacteria that's never been cultured before. Other bacteria should not be able to grow in it, because otherwise you still got that problem.
And it's amazing that within the human gut, we have an almost infinite number of perfect environments for bacteria.
Yeah, and so what happens is that you and I might have the same environment that's occupied by a different bacterium. So there's a finite number of environments, and there's a bigger number of bacteria that occupy those environments. So all those interactions between them dictate which one wins out in different people.
Right.
So that's part of that becomes part of the problem in defining your healthy microbiome, because it's not unhealthy just because you don't have bacterium A and you've got bacterium B instead.
It's individual. I mean, when Prof Flint was on, he talked about, he pushed back a little bit on, you need this bacteria there, rather you need the groups, the functional groups.
Exactly. So the function of the bacteria is more important. It's not the who's there, it's what they're doing. That's really important.
Right. And are our tools that allow us to measure what they're doing getting better, Or do we still have a lot to do to be able to profile that?
Yeah, I think we can. Well, it depends which part of what they're doing you're talking about. So in terms of what they themselves are doing in terms of degrading substrates and producing metabolites, we know what they're doing. But how those metabolites then interact with the human host, that's something that we're still probably maybe halfway through understanding. We know a lot more than we did 5 years ago or even a year ago, but there's still a lot to learn.
What are you most excited about in the context of your own research at the moment?
I think at the moment, it probably depends which week you ask me actually, which particular research project's at the top of my brain at the time. But yeah, at the moment I would say I am particularly interested and excited about the fact that we might be discovering bacteria that can very specifically stop other bacteria growing and avoid the use of antimicrobials, because I think some of the, and I'm not just talking human medicine here, I'm talking antimicrobials in animal husbandry as well, because that also is a big deal in terms of antimicrobial resistance. It's going back to my ruminant days as well, I guess.
Yep. And your hope is that classical antimicrobials take a back seat at some point and we've got a different way of doing it.
Yeah, and I think we do need to think about a different way of doing it. I mean, we've already realised in the hospital setting that actual basic hygiene can help instead of antimicrobials in certain ways. And it's the same on the farm. Like, just in thinking about the farm environment in a different way can actually save using antimicrobials.
Do you take any probiotics?
That's a very good question. I do sometimes. I would say when I'm not a huge yogurt fan, but I do like a little spoon of yogurt in my cereal in the morning, and I would always have a live bioactive yogurt in the morning. I don't take any probiotic capsules unless I've been taking antibiotics.
There we go. Thank you for sharing
then
that.
I would take probiotics.
Thank you for sharing that. And I won't ask you to recommend any particular brand.
No, please don't.
I had someone else on a couple of weeks ago who gave a really good overview of what to look for to find a good probiotic. In terms of, looking for studies on the internet. Do they tell you the dose? Do they name the specific strain rather than just saying it contains X number of live microorganisms? So that will come out before this one comes out, actually, so the listeners will have been primed with that. I'm looking at the time, and I know you have things to do after the podcast. We've covered everything, actually, that I wanted to cover.
Okay, so just to reiterate what you were saying about probiotics there, having the evidence base is really important. So probiotics are a type of bacteria, and we've said about how there are thousands of different bacteria, so not all probiotics are equal. So the person that you had on that said you have to really look at the evidence and take what you want for the specific purpose that you want it for is really important. The caveat to that would be if you had been on antibiotics, when you really aren't looking for a specific purpose of the probiotic, you're just trying to make sure that you don't get any detrimental bacteria taking over whilst your own bacterial load's been decreased, then probably that's the time when maybe it doesn't matter quite so much which one you take.
Okay.
So in terms of the asking you a question.
Well, you've been asking me a couple, so I thought, why not, if you'd like.
I don't know. Nah, it's okay. I'll let you off the hook this time.
Good. Finally, what are your thoughts on FMT?
Yeah, so I think FMT has been really effective in Clostridioides difficile. It's really helped a lot of people. And I think for that, and it's been, as well as I do, that it's got approval for use against Clostridioides difficile.
Yes.
I think I am concerned about under-the-counter treatments with FMT for everything, for obesity, for lots of random things. The FMT seems to be, in some places, being held up as a sort of cure-all, and I really don't think that's safe, and I don't think it's going to be effective. And I think if anybody was thinking about getting FMT, then you really have to make sure that you're going to a supplier that's got lots of evidence-based safety, because I mean, we have touched on how diverse the microbiota is. We were talking about the bacteria. We didn't even talk about the viruses and everything else. And so if you don't know something's there, you can't screen for it. So there's a lot of caveats, I would say, with FMT still.
We position ourselves like completely away from traditional FMT because, I mean, if you were to break open an EBX capsule, you'd see a powder which was dry, off-white in colour, and odourless. And it's consistent because of the manufacturing process and donor screening processes. So we're actually saying, this is something totally different. This is a next generation.
Yeah.
The same principles still apply because it's biologically sourced starting material. Essentially, it's come from a person. Now, we've heard about all the amazing things about the human large intestine today. It's an incredibly efficient fermenter system. It's home to a very wide diversity of potentially therapeutically active bugs that could help somebody in need. The flip, though, obviously, is that people do carry potentially pathogenic organisms, and therefore it's fundamental, it's critical to the longer term success and safety and scalability of products like ours to have as robust screening as possible. There's never going to be no risk.
Well, that's true. That's true of everything, isn't there?
But it's about risk mitigation, and then once you've exhausted your risk mitigation to the point where it's, I think, at the verge of being what is just absolutely possible, then it's about the risk benefit assessment and the patients, and
that's exactly right. So then your Clostridioides difficile, when your patient is going to die or get FMT, which is 99% successful against C. difficile.
Yeah, it varies, but
97. It's really high.
It's very high, yeah.
I would take it. If I would take it. If I was that person, I would take it. And in that situation, because the risk benefit is so big.
Yeah.
But for somebody who's thinking it's going to actually help them to lose weight, I would say change your diet first.
I mean, someone might be thinking, this changing my diet stuff, it's just so hard and I don't like it. Can I just take one of antibiotics capsules every day to get my diversity?
Well, we've mentioned this. So those bacteria you take in are never going to survive unless you feed them properly. So if you want your microbiota to be a fibre-loving microbiota, then you're going to have to eat the fibre.
Do you think it might be possible with the AI and everything to just create a bar that contains all the beneficial fibres you could ever want, and it tastes good, tastes like...
You're not going to be able to pull that out of the ground. That's going to be your highly refined food that we've said is really bad for you.
Yeah, no, you're right. I get all these ideas all the time.
And it's boring. Nobody wants to eat a single bar. That's like space age sort of take your little pill and that's your dinner.
Yeah.
People don't want to do that.
Well, hold on a second. People like me want to develop that, because I'm being serious. I that's the kind of world.
You hate cooking that much.
I love cooking. Ask anybody who has been wined and dined by me. I put a lot of effort into the cooking.
Why do you want to eat a pill?
So to be absolutely clear, I do not want to eat a pill. Okay. However, I say people like me, these entrepreneurial evangelist type people who think impossible is nothing and who want to make the world better. I look at the world and I think there has never been better availability to fruit and vegetables ever, full stop. There is so much excess food in this country that food waste is one of our biggest problems, right? And we still don't have people eating the minimum fibre targets. So we've got all the food. The education piece is kind of there. How do we fix that? So one solution is we need to educate people better, and we must educate the kids. We must educate everybody as best we can. And I believe that if people change their diet, they get their cravings for their fruit. They feel good. Skin looks good. Everyone's commenting, "Wow, you look really well," and they just do it. If you can get somebody over that hump, they become a bit of a fruit evangelist. But my thinking is, it's not realistic to expect everyone to have their fruit and veg. So why don't we, at a population level, through all the technology we have, shoot for the stars and achieve the space age goal of, yes, you just take this James McElroy pill and you will get your fibre target of 50 grams of all the various different types of fibres, pectin, inulin, the starches, everything is all in there.
Well, you need to put your protein in there as well, and your fat, and all the other components of your healthy diet.
One step at a time. No, I like to think about these thought experiments, but in reality, you're absolutely right. Just eat your fruit, veg, and
eat a healthy, diverse
fibres,
diet.
And we've talked about
cereal fibre too. Remember,
cereal fibre. That is a key take home today for sure. You've really pushed that, and I think that's great. Cereal and nuts and seeds, all that kind of stuff.
All those different types of fibre.
All good stuff. So I'll conclude by asking you one more question. Where do you see the field going in the next 5 years?
In the next 5 years, the microbiome field.
Yes.
Well, hopefully in 5 years' time, we will be at a position where we'll be able to say to people, if you eat this type of fibre, you will encourage the growth of this bacterium and it will produce this product that will improve your health. And we are sort of almost there with some things, but that would be an achievable goal in 5 years, I think.
Karen, it's been an absolute pleasure.
Thank you.
A lot of fun. Hope you did too.
Enjoyed.
And thanks so much for coming on. Really appreciate it.
Thank you.
Cheers.
Bye.